Hi, let us now examine how amino acids are grouped together in a complete protein. So, X-ray crystallographic and nuclear magnetic resonance, is short for we call it NMR studies, have revealed the detailed three-dimensional structures of thousands of proteins. We begin here with an examination of myoglobin, the first protein to be seen in atomic detail. Myoglobin, the oxygen storage protein in muscle, is a single polypeptide chain of 153 amino acids. The capacity of myoglobin to bind oxygen depends on the presence of heme, a non-polypeptide prosthetic, we call it a helper group, consisting of protoforwarding IX and a certain RN item. Myoglobin is an extremely compact molecule. The folding of the main chain of myoglobin, like that of most other proteins, is complex and devoid of symmetry. The overall course of the polypeptide chain of a protein is referred to as its tertiary structure. A unifying principle emerges from the distribution of side chains. Strikingly, the interior consists almost entirely of non-polar residues such as leucine, valine, methionine, and phenylalanine. Charged residues such as aspartate, glutamate, lysine, and arginine are absent from the inside of myoglobin. The only polar residues inside are two histidine residues, which play critical roles in binding iron and oxygen. The outside of myoglobin, on the other hand, consists of both polar and nonpolar residues. The space-filling model shows that there is very little empty space inside. Some proteins that span biological membranes have the reverse distribution of hydrophobic and hydrophilic amino acids. For example, consider porins, proteins found in the outer membranes of many bacteria. So membranes are built largely of hydrophobic alkyne chains. Thus, porins are converted on the outside largely with hydrophobic residues that interact with the neighboring alkaline chains. In contrast, the center of the protein contains many charged and polar amino acids that surround a water-filled channel running through the middle of the protein. Thus, because porins function in hydrophobic environments, they are inside out, relative to proteins that function in aqueous solution. Certain combinations of second structure are present in many proteins and frequently exhibit similar functions. These combinations are called motifs or supersecondary structures. For example, an alpha helix separated from another alpha helix by a turn we call an alpha helix turn helix unit, is found in many proteins that bind DNA. Some polypeptide chains fold into two or more compact regions called domains, range in size from about 30 to 400 amino acid residues. For example, the extracellular part of CD4, a protein on the surface of certain cells of the immune system, comprises four similar domains of approximately 100 amino acids each. Proteins may have domains in common even if their overall tertiary structures are different. So far, we have considered three kinds of structures. And primary structure is the amino acid sequence. Secondary structure refers to the spatial arrangement of amino acid residues that are nearby in the sequence. Some of these arrangements give rise to periodic structures. The alpha helix and the beta strand are elements of secondary structure. Tertiary structure refers to the spatial arrangement of amino acid residues that are far apart in the sequence and to the pattern of disulfide bonds. We now turn to proteins containing more than one polypeptide chain. Such proteins exhibit a fourth level of structural organization. Each polypeptide chain in such a protein is called subunit. Quaternary structure refers to the spatial arrangement of subunits and the nature of their interaction. The simplest sort of quaternary structure is a dimer, consisting of two identical subunits. This organization is present in the DNA-binding protein, like found in a bacteria, virus we call it like lambda. More complicated quaternary structures also are common. More than one type of subunit can be present, often in variable numbers. For example, human hemoglobin, the oxygen-carrying protein in blood, consists of two subunits of one type, designated as alpha, and two subunits of another type, designated as beta, as illustrated in the figures shown here. Thus, the hemoglobin molecule exists as an alpha-2, beta-2 tetramer. Subtle change in the arrangement of subunits within the hemoglobin molecule allow it to carry oxygen from the lungs to tissues with great efficiency. Virus makes the most of a limited amount of genetic information by forming codes that use the same kind of subunit repetitively in a symmetric array. So the code of, let's say, a rhinovirus, the virus that causes the common code, and it includes 60 copies of each of 4 subunits. The subunits come together to form a nearly spherical shell that encloses the viral genome. The classical work of Christian Efferson in the 1950s on the enzyme ribonucleases revealed the relation between the amino acid sequence of a protein and its conformation. So ribonucleases is a single polypeptide chain consisting of 124 amino acid residues cross-linked by 4 disulfide bonds. Efferson's plan was to destroy the 3-dimensional structure of the enzyme and to then determine what conditions were required to restore the structure. So agents such as urea or guanidinium chloride effectively can disrupt a protein's non-covalent bonds. Although the mechanism of action of these reagents is not fully understood, computer simulations suggest that they replace water as the molecule solventing the protein and are then able to disrupt the van der Waals interactions, stabilizing the protein structure. So the diphosphide bonds can be cleaved reversibly by reducing them with a reagent such as beta-lactoethanol, and in the presence of a large excess of this kind of reagent, the disulfide are fully converted into sulfur hydrolase. Most polypeptide chains devoid of cross-links assume a random coil conformation in 8 molar urea or 6 molar guanidinium chloride. When ribonucleases was treated with beta-lactoethanol in 8 molar urea, the product was a fully reduced, randomly coiled polypeptide chain devoid of enzymatic activity. So when a protein is converted into a randomly coiled peptide without its normal activity, it is said to be denatured. A quite different result was obtained when reduced ribonuclease was reoxidized while it was still in 8 molar urea, and the preparation was then dialyzed to remove the urea. So ribonucleases reoxidized in this way had only 1% of the enzymatic activity of the native protein. Because there are 105 different ways of pairing 8 cysteine molecules to form 4 diphosides, only one of these combinations is enzymatically active. So the 104 wrong pairings have been, pictures equally termed scrambled ribonuclease. So Afniston found that scrambled ribonuclease spontaneously converted into full active native ribonuclease when trace amounts of beta and maltodextrin were added to an aqueous solution of the protein. The added beta-maltodextrin catalyzed the arrangement of diphosphate pairings until the native structure was regained in about 10 hours. This process was driven by the decrease in free energy as the scrambled conformations were converted into the stable native conformation of the enzyme. So what determined whether a particular sequence in a protein forms an alpha helix or a beta strand or a turn? One source of insight is to examine the frequency of occurrence of particular amino acid residues in these secondary structures. So residues such as alanine, glutamate, and leucine tend to be present in alpha helix, whereas valine and isoleucine tend to be present in beta strands, while glycine, asparagine, and proline are more commonly observed in turns. Can we predict the secondary structure of protein by using this knowledge of the conformational preferences of amino acid residues? Accurate predictions of secondary structure adopted by even a short stretch of residues have proved to be difficult. Note that the conformational preferences of amino acid residues are not tipped all the way to one structure. For example, glutamate, one of the strongest helix formers, prefers alpha helix to beta strand by only a factor of 3. So the preference ratios of most other residues are smaller. Indeed, some pentra or hexapeptide sequences have been found to adopt one structure in one protein and entirely different structure in another. So hence, some amino acid sequences do not ubiquitously determine secondary structure. Tertiary interactions between residues that are far apart in the sequence. It may be decisive in specifying the secondary structure of some segments. So context is often crucial. So the conformation of a protein has evolved to work in a particular environment. Proteins can be denatured by any treatment that disrupts the weak bonds stabilizing tertiary structure, such as heating, or by chemical denatures such as urea or graphene chloride. For many proteins, a comparison of the degree of unfolding as a concentration of denaturant increases revealed a sharp transition from the folded or native form to the unfolded or denatured form, suggesting that only these two conformational states are present to any significant extent. The sharp transition seen in this figure shows that protein folding and unfolding is an all or none process that results from a cooperative transition. The consequences of cooperative folding can be illustrated by considering the contents of a protein solution under conditions corresponding to the middle of the transition between the folded and the unfolded forms. Under these conditions, the protein is half-folded, yet the solution will appear to have no partly folded molecules, but instead look as if it is a half-half mixture of fully folded and fully unfolded molecules. Although the protein may appear to behave as if it exists in only two states, this simple two-state existence is an impossibility. So at a molecule level, even simple reactions go through reaction intermediates, and so complex molecules such as protein cannot simply switch from a completely unfolded state to the native state in one step. So although figures suggest that a discrete pathway, each of the intermediates shown represents an ensemble of similar structures, and thus a protein follows a general rather than a precise pathway in its transition from the unfolded to the native state. The energy surface for the overall process of protein folding can be visualized as a funnel. So the wide rim of the funnel represents the wide range of structures assembled to the assemble of denatured protein molecules. So as the free energy of the population of protein molecules decreases, the protein moves down into a narrower part of the funnel and fewer conformations are accessible. So at the bottom of the funnel is the folded state. With its well-defined conformation, many parts can lead to the same energy minimum. Proteins are able to perform numerous functions that rely solely on the versatility of their 20 amino acids. In addition, many proteins are covalently modified, though the attachment of groups other than amino acids to augment their functions. So the preceding modifications consist of the addition of special groups to amino acids. Other special groups are generated by chemical rearrangements of side chain and sometimes the peptide backbone. For example, the jellyfish produces green fluorescent protein, short for GFP, and this protein which emits green light when stimulated with blue light. Okay, so the source of the fluorescence is a group formed by spontaneous rearrangement and oxidation of the sequence serine, tyrosine, glycine within the center of the protein. Since the discovery of GFP, a number of mutants have been engineered which absorb and emit light across the entire reversible spectrum. So these proteins are of great utility to our researchers as the markers within the cells. Okay, so that's all, and thank you for your attention.